Okay, so hi everyone. Welcome to our Summer of Science. Today we have a great presentation. Let me just pull up my slides for a second. And I know that some of you have already started to do this, which is fantastic. Appreciate that you've done that. So if you haven't, just go ahead, introduce yourself, who you are, where you're from. We love to just create a nice community here. I'm Lauren Traster. I'm the 4-H Teen and Leadership Program Coordinator here in Vermont. And one of the programs that I run are these science cafes. I know many of you have been here before, so that's great. So you know our protocols, but I always go over them. So when we gather together here in Zoom land, all our participants are muted today, but you can share your thoughts via the chat box. Keep your chatting to the topic. We like that the chat box is related and about what our presenter is talking about. Maybe you're responding to a question she asks you. It's really not meant to be a distraction to everybody else, so make sure you use it appropriately. If you have a question for our presenter today, we use a separate tool called the Q&A box. So you can load your questions in there as they come up, and our presenter will answer your questions as it fits into her presentation. But don't worry, all questions will get answered before we leave today. If you like a question someone else asked, or if it was the same question you were planning on asking, there is a mechanism in there that you just click the thumbs up, and that actually lets us know that more than one person is interested in that question. It actually moves that question up the ranking. But again, we are going to answer all questions today. All questions are good questions, so just remember that. Be courteous and respectful. Again, no distractions, and in this case, you can see us, but we can't see you, so there won't be any video distractions. It really just comes down to how you use that chat box. So we ask that you stay engaged, participate fully today. And again, if you do need our live streaming services for closed caption, I've already put the link in the chat box. And all you have to do is click on the hyperlink, and that will take you to a third-party site, and we have someone who is doing the closed captioning today. So, moving on, I do want to mention, so most of you know that we have this whole series going on this summer called Summer of Science. Today is our third session in the series, but I want to point out that next week, we actually are overlapping with another program called the Natural Resource Management Academy. So that program runs Monday through Friday next week, kind of the same time, 1 to 2:30. The Wednesday session is going to overlap, so it's for both Natural Resource Management Academy and Summer of Science. What I'm hoping is, because the presentations do function very similar to what we're doing today, but we're going to do a little bit more. We're going to have some fun icebreakers and some wrap up, like we're going to do trivia at the end of the week. So it's a nice series if you're interested in natural resource management. But if you only want to come on Wednesday next week, you can. You sign up for the whole program, but you'll get a link for Wednesday. So I just wanted to mention that, point that out. The website at the bottom of this page is where you would go to register. So hopefully you'll join us next week. And if not, we go back to, I am all over the place today. We go back to just our traditional Summer of Science on July 22nd. And that topic is a different kind of veterinarian. We have a specialist in veterinary infection disease epidemiology. It is going to be really interesting. So, and then we continue on throughout the summer. As you can see, the dates and the topics. It should be really fun. So hopefully you guys will keep joining us. So I do want to introduce our presenter today. Our topic is, what do pickles, yogurt, and soy sauce have in common? How fermentation feeds us. Our presenter is Emily, and I was ahead of time was going to ask you how to properly pronounce your last name. Is it Forauer?
Yeah. Yep.
I did it. So Emily is a master's degree candidate in the Department of Nutrition and Food Sciences at the University of Vermont, studying under the direction of Dr. Andrea Etter, who I should point out is doing her own Summer of Science session in August on backyard chickens. Yeah, and salmon and salmonella. So you'll be able to hear from Emily's mentor and grad student later on this summer. So she works with Dr. Andrea Etter and a graduate teaching assistant for the food microbiology and food technology courses in that department. She is currently studying the resistance of mature Listeria. Oh my God, you're giving me Latin words. Monocytogenes?
Close enough. It works.
Here's a lesson to all of you out there. Normally ahead of time I practice and I learn how to pronounce these hard Latin science words I did not get to today. So practice ahead of time. So anyway, she studies the resistance of mature Listeria monocytogenes, biofilms to sanitizers, and leading a project to determine the presence of pathogens and probiotics in microbial communities isolated from raw milk from homestead dairy producers. So this is going to be a great topic. I'm going to unshare my screen. Emily, I'm turning it over to you. And for those of you who just joined us, if you haven't given a hello in the chat box, go ahead and let us know where you're from. And we'll give our attention to Emily now.
All right, let me just share my screen. All right, hang on a second. I think something else is popping up.
Now, what you sent me the other day, was that complete? Because I could get it off my computer. Emily, you're on mute now. Oh no, guys, we may have jinxed this. She has been having trouble with Zoom all day. Emily, if you can hear me, you are on mute. Let's see.
All right.
Oh, now I can hear you.
You lost me for a second, but I'm back.
Yeah, you were on mute, and now there's two screens of you, one that's on mute and one that's not. We don't see you, but that's okay, but we don't see your screen.
All right, I think that I'm technically an attendee right now. Let me see.
Oh, you came in. Okay, okay, you're working off of two, so hang on a second. Let me make both of you co-hosts. Did you all know that our presenter apparently now she's dividing? Okay, you should be able to now.
Okay, share my screen. Yes, okay. Okay, can you see it?
Oh, I see me.
Okay, that's the screen. That's the screen.
Okay.
All right.
There we go.
Now we're better.
Good. Yes.
Okay, awesome. All right.
And again, all of you out there, thank you for your patience with some of our technical difficulties today.
Yes, thank you so much. This is my first time presenting on Zoom. So welcome to What Do Pickles, Yogurt, and Soy Sauce Have in Common? How Fermentation Feeds Us. So just a little bit more about me. I went to the University of Connecticut as an undergraduate where I found my love for food microbiology. This is a picture of me there. I am pouring some enzyme into a cheese vat. So what I did there primarily was working with queso fresco and milk and Listeria monocytogenes to see how different products could kind of deter pathogen growth in those foods. After I graduated from UConn, I went to Chicago for a year where I worked for the nonprofit organization Stop Foodborne Illness. This is an organization that works with people who were sick from food, advocates for stronger government policies surrounding food safety, and shares people's stories with industry so they know that what they're really doing in food processing is super important to people and public health. And now I am at the University of Vermont in the Etter Food Microbiology Lab, working with Listeria monocytogenes. So these are some of my Listeria strains that I work with. And this is a pretty recent photo of me, note the mask, working with the biofilms that I study. So an outline for this presentation, we're going to go through a little bit of an introduction of microorganisms in food, talk about the factors that affect microbial growth, talk about what fermentation is, and then go through some fermented foods specifically. These are by all means not an extensive list. These are just a few, some of my favorites that I personally like to eat. And then we'll talk about probiotics and how you can do a little bit of an edible microbiology lab at home. And if we still have time, we'll use the comments to do some food microbiology Jeopardy. So microorganisms are everywhere. They're in obvious places like soil and dirt, but they're also in not so obvious places like the air. They're on basically every surface you can imagine, including in your body. So this right here is a little diagram showing that even across your own body, there is incredible diversity in microbial communities. So someplace like the bottom of your foot is not going to have the same microbes that the inside of your mouth does. And that's because there are different microenvironments. There's different levels of salt, moisture, oxygen availability. So all of these different pie charts are showing that even across one person, the microorganisms are so very different. And then this little comic references the Human Microbiome Project, which is a project run by the NIH that is seeking to categorize the human microbiome in its entirety. Studying the normal human microbiome can help us better characterize dysbiosis, which is when you're sick because your microbiome is out of whack, which is very interesting. I've linked that in the resources if you guys are interested. So microorganisms are everywhere. That also means that they're in the foods that we eat. So in things like produce, things that you pick off of a tree yourself, or something that you buy at the grocery store, there's going to be microorganisms. Dry foods like rice, beans. The one exception is canned foods. Those are technically sterilized. And so when you have microbial growth in canned food, that's really not good. That's generally botulism, which we don't want to eat at. That's why you don't buy bulging cans. So when you have food and microbes, what do you get? Well, yeast, mold, bacteria, and viruses all are on food, and they all fall into three categories. These are just the general categories. It's from a really old movie. I've never seen it, but we call the microbes that are on foods the good, the bad, and the ugly. So good microorganisms will improve, preserve, and protect the food that we eat. So things that we'll talk about today are the good. So that's things like lactic acid bacteria, yeasts, and molds that will ferment things. They'll outcompete pathogens. They'll increase the shelf life of a product, and also they can make food taste better. The bad are pathogenic. They cause illness. So they'll make you sick. These are organisms like Listeria, like E. coli, I'm sure you all have heard of it, and Salmonella, which you guys can hear more about in Dr. Etter's presentation later. So these will be on food, you'll ingest them, and they'll multiply within you, causing nausea, diarrhea, vomiting, all of those bad things that we associate with foodborne illness. These are also the ones that you hear about a lot. So around Thanksgiving in 2018, there was a romaine lettuce recall because of E. coli. There are other recalls due to other things like labeling, but when you hear of one because of a microorganism, they're the bad ones. And the ugly, these are spoilage microorganisms. So they're not necessarily going to make you sick, but they're going to make food gross or not edible. Something like mold that grows on bread that you leave in your lunchbox, that I know I still do, and mold on berries, slimy yeast on potatoes if you store them improperly and they rot. These are things that generally need time, need some way to outcompete the natural good microorganisms. So like on berries, if you wash away the good microorganisms, the bad ones have an opportunity to thrive.
Hey, Emily.
Yeah.
Alex is asking, does romaine lettuce still carry E. coli?
Not anymore. There are currently not any active recalls on that. It grows in the ground. There's always going to be that risk. That's why we wash produce. But yeah, romaine lettuce is currently safe. It has been safe for a very long time since. If you want to stay up to date on recalls like that, following USDA and FDA announcements will let you know what's going on in our area. Cool. So, FATTOM is not an insult, I promise. These are the conditions that we think about when talking about microbes and whether or not they're going to thrive. So, food is all of the nutrients that are present that are available for energy and growth. So, something like sugar needs to be present for basic cellular energy, and more complicated structures like proteins and lipids are going to be really important for cell division, making essential cell structures like the cell wall, amino acids to form proteins for regular cellular function. All of these are very important, and each microbe needs its own kind of balance of these things in order to thrive. So acid is next. This is the pH scale. It goes from 1 to 14, and it is, at its essence, the availability of hydrogen ions. So as you get more acidic, closer to 0, there are more hydrogen ions available to basically react with molecules and attack essential cell structures. And if that happens, the cell will die. Different organisms have evolved to thrive at different pHs. So we are about a little bit more acidic, a little bit more basic than neutral, depending on what bodily fluid you're talking about. That's where human pathogens are best adapted. They don't do really well in super basic or super acidic environments, but there are exceptions to everything. There are bacteria that live in deep sea vents, so those are adjusted to the more basic pH of seawater, and things that can live in kombucha that's more acidic, those are more adapted to having a higher concentration of hydrogen ions around. Next is temperature. So this is kind of a Goldilocks situation of microorganisms. Doesn't want it to be too hot, doesn't want to be too cold. You'd want the just right temperature. Again, mesophiles, that means bacteria that grow at middle temperatures. Like human pathogens will grow at about human body temperature, a little higher, a little lower. And there are also thermophiles that grow where it's really hot, and saprophiles, like Listeria, can grow in the refrigerator. So even if the temperature is colder, they can still grow. Bacteria do not die in the freezer, fun fact. They just slow down their growth so far because the temperature slows down all of those metabolic reactions. So if you freeze something and then thaw it, there can still be bacteria in it. That's why we cook things like we cook chicken to 165 degrees Fahrenheit to kill all of the bacteria, because again, those pathogens are adjusted to the middle temperatures, and that high temperature of 165, they can't survive. So next is time. Time is essential for cell division. So the kind of half-life, if you will, of E. coli is 30 minutes. So for binary fission to occur, which is the method of asexual replication of bacteria, that's 30 minutes long. So the longer you have, the more bacteria you'll have. And that's really important when we think about fermentation, if we want enough time for these bacteria to do the good things that we want them to. Oxygen. Microorganisms need different oxygen concentrations to survive. We'll talk about the effect of oxygen on fermentation next, but there are different things like obligate anaerobes that definitely need an anaerobic without oxygen environment, obligate aerobes that definitely need oxygen to survive, and human pathogens are facultative anaerobes. So that means they can grow in oxygen or without oxygen. They can survive in either. And finally is moisture. Microorganisms all need water for essential functions. Some have evolved to use more or less, like aquatic bacteria. They can survive in conditions where the concentration of ions outside of their little cell bodies is less than inside. Some organisms can evolve to sporulate. So when there's not enough water they will form like basically a time capsule of DNA. And when there is moisture again, that will activate and the bacteria can grow again happily. So it is time for our first poll. Which of these 6 factors do you think would be the most essential for microbial growth in foods?
Okay, so I'm about to launch the poll. Your choices are food, acid, temperature, time, oxygen, or moisture. The poll is being launched.
All right.
If for some reason you don't see the poll, you can type in the chat box. Otherwise, put your answer into the poll. We'll see what you guys think.
Let's see the poll.
Let's see, we'll get a couple more answers in there. It looks like the majority is going with food, with temperature being a close second.
All right, awesome.
Share those results.
Excellent. So, great news, you're all right. It's really, yeah, trick question. All of these are important to balance. So different microorganisms definitely need certain foods. So there are certain bacteria that can't synthesize their own vitamin B. So when I grow them in the lab, I need to supplement with yeast extract. If you guys have ever eaten nutritional yeast yourself, you know that's a good source of vitamins. It's also a good source of vitamins for bacteria. So food is super important, especially if the bacteria lacks the cellular machinery to make things themselves. And again, all of the other ones are super important as well. Every microbe is evolved differently. Everything has its own kind of exception to the rule. But yeah, so if you're interested in more, I could talk forever, but we'll move on to what is fermentation. So fermentation is an anaerobic process which converts complex organic molecules into simpler molecules and produces chemical energy. So this is the most basic definition of fermentation. There are also exceptions. Again, yeast can ferment in an aerobic environment. Bacteria will generally only ferment in an anaerobic environment without oxygen. So aerobic respiration has oxygen as the final electron acceptor of the electron transport chain. So in this cellular process, you start up here at glycolysis. You have some glucose or other food. You undergo this process. You produce some ATP, that's chemical energy for the cell. Then you enter the mitochondria, powerhouse of the cell, and you go through the Krebs cycle and then the electron transport chain. So this chain makes the most energy of respiration, but it needs oxygen to be the electron acceptor to form water as a byproduct. When oxygen isn't present, the cells undergo fermentation. So these chemical reactions are much more complicated, so they're simplified in an arrow. But basically, oxygen is not present, so other molecules need to be that electron acceptor, and you end up with ethanol and lactate. In different proportions, different cells produce different amounts. They'll have their preferred chemical pathway, but this is what you end up with, right? Metabolism is very, very complicated. When we talk about fermentation, there are a lot of other metabolic processes going on. But this is just very, very briefly what fermentation is. You have an anaerobic environment and you end up with ethanol or lactic acid. Most of what we're talking about, we're going to be dealing with lactic acid. So, fermented foods, finally. Yeast, mold, and bacteria can all ferment things. I believe that I mentioned that viruses are found in foods. They're part of the microorganisms that are found everywhere, but viruses cannot replicate without a host, so they do not ferment. They can't replicate by themselves, they don't do it. The transformation can be in flavor, texture, nutrient content, and definitely shelf stability is a really important So on the left here, we have bacteria. These are what we'll say are some lactobacillus and lactococci. Those are very common genuses of lactic acid bacteria. In the middle here, we have yeast. Yeast has this budding morphology where you can see all of the cells are kind of like Easter egged next to each other. The bacteria are also in chains, but the yeast have this more ovate shape. And they're also much bigger under the microscope. These are not to scale. You definitely be able to tell under a microscope. And molds, I could not find a very good picture of under a microscope, but they are filamentous, they're fluffy, and you can identify them that way. So the foods that you can ferment range from milk, which you can get yogurt, cheese, and sour cream from, 3 totally different products. You can turn cucumbers into pickles, peppers into hot sauce, and soybeans into soy sauce. So there's very obviously a very big range here, and we'll talk a bit about all of these. So first is pickles. Pickles are cucumbers that are stored in a brine solution. So this brine solution is an anaerobic environment with high salt that discourages unwanted bacteria. So those unwanted bacteria are competitors that will outcompete the lactic acid bacteria that we want. And those lactic acid bacteria will ferment the natural sugars. I know that we don't usually think of cucumbers as a sweet food, but they're there. And so starter cultures are not necessary for pickles. These lactic acid bacteria are naturally occurring. They're everywhere. They're on your skin. They're on fruits. They're on vegetables. So we're really just taking advantage here of what microorganisms exist naturally in our environment to improve our food. The lactic acid bacteria will produce lactic acid and drop the pH from around neutral but salty to 3.4 to 3.6 within the following weeks. And this is really important because it will discourage pathogen growth. This is really what's making the pickles safe. They're what's prolonging the shelf life of the cucumber. And that's kind of what we need to consider when considering safety, when considering what the fermentation is actually going to achieve. Fermentation is complete when the bubbling stops. So as we saw in the diagram before, carbon dioxide is a byproduct of fermentation. And when that bubbling stops, the lactic acid bacteria have done their jobs. That's when you can can the pickles and basically extend their shelf life indefinitely if you do everything right. There are also lacto-fermented hot sauces. So basically the point here is that both vegetables are fermented the same way. You take advantage of what's already naturally occurring on these vegetables, fruits, and there's lactic acid bacteria everywhere, and you can transform something into something even more delicious if you like spicy food or pickles. I don't know if you guys can see my I Heart Pickles shirt, but I thought it would be appropriate to wear today. So next we'll talk about fermented dairy foods. So fermented dairy foods all start with milk. Then you add a little bit of culture, you wait some time, and you'll either end up with something like yogurt or you'll end up with cheese. Now, what's the difference? What would change between these two things? We started with milk, we added a culture, we waited time. The difference really is the culture. So here we're going to talk about some highlighted organisms that really make a difference in what we're going to get out of our raw product. So both yogurt and cheese need to start with milk and added culture. So unlike pickles, you want to add these cultures in. So these are some common ones in yogurt: S. thermophilus, L. bulgaricus, that's lactobacillus, and some others that will drop the pH slowly and coagulate the milk proteins to make that kind of semi-solid yogurt texture. So here's just some pictures of yogurt. This is a giant microbe of lactobacillus that is in yogurt. This is the Chobani label, actually. So certain yogurts will actually list the strains of bacteria that they use as live cultures. Some don't, but I'll definitely have them. And then with cheese, you have very obviously different cultures. So the first ones here are molds, the middle one is a yeast, and then there are more bacteria, including some lactic acid bacteria. So the lactic acid bacteria will form the curd by dropping the pH quickly, so those proteins will precipitate, which will be the curd, and then the whey is the rest that separates off. And then the other microorganisms will develop texture, develop flavor, and the processes for making cheese are also incredibly varied. We will not get into that. But you can buy these cultures from manufacturers. They come in these little packets, they're dehydrated, and you just add them to milk, depending on what cheese you want to make, and you can get all kinds of different outcomes. So I wanted to highlight the yeast that is on these so-called brainy cheeses. That's Geotrichum candidum. It's soft, it's kind of wrinkly, and it also breaks down proteins, breaks down fats to develop the rind and the texture and the flavor of the cheese. The mold that I would like to highlight is Penicillium roqueforti, which is in blue cheese. This is actually Bailey Hazen Blue from Jasper Hill, so local, cool. And this is a picture of the mold under the microscope. Again, that's breaking down proteins, breaking down lipids, developing the flavor primarily in blue cheese. If anybody's never had blue cheese, it's very pungent, easily identifiable, and that's definitely from the mold. And the last one that I'd like to highlight is in Swiss cheese. This is Propionibacterium freudenreichi, and this lactic acid bacteria actually produces a lot more carbon dioxide that forms those characteristic eye holes in the Swiss cheese and other Alpine style cheeses. Something else that I wanted to mention, in cheeses, there are certain styles like a hard cheddar that can process the sugars in the milk. So generally speaking, aged cheddars will have less lactose in them because of the bacteria that are active over a long period of time and produce lactase and break that down. So that's cheese. And these are both lactic acid fermented foods, again, like the pickles. So moving on to kombucha.
Yeah. We have a question that just came in. Who first figured out the process of fermentation at the microbe level? And I also want to point out, your video is off. And when you mentioned your shirt, I didn't know if you knew your video was off, if you need it off.
It's okay. I've been gesturing with my hands a lot, but my shirt says I heart pickles. That was the whole thing.
Okay. I don't want to mess with the technology, so we don't have to get to the phone. But I figured we should, while you're talking about this fermentation at the moment, if you could answer that question.
Absolutely. So I will tell all of you that if a scientist says that they know everything or is hesitant to say that they don't know, they're not a very good scientist. And I will tell you that I do not know who first discovered fermentation. Fermentation actually dates back in all cultures that I can think of off the top of my head, basically because it is such a good way to preserve food, much like drying out meats, salting fish. Fermentation is a way to prolong a harvest. Like if you harvest milk one day, you can have cheese for many weeks after. So I'm absolutely not sure. I hope that it definitely doesn't answer your question, but fermentation is an ancient practice for sure.
Sounds like a good research question after we end today.
Yeah, for sure. All right. Now we'll talk about kombucha, which is an ancient beverage from northern China. So kombucha is a really good example of symbiosis. That's when microorganisms or big organisms work together to form a community. So the basic recipe is very simple. It's a two-step fermentation with black tea, sugar, and the SCOBY, or the mother, in an open air vessel. So you probably have all these things in your house except for a SCOBY, but you can make one, which I'll talk about a little bit later. So this is what kombucha looks like while it's doing its thing. It's fermenting. This is the first step of kombucha production. Then you bottle it without the SCOBY and put it into a closed vessel, and that's what allows the fermented tea to carbonate, not because of a difference in the microbes, but because now that there is pressure, that gas is going to be forced to be into the liquid. So there's not really like a different addition of culture. It's just the vessel that it's in. So the SCOBY I want to talk a little bit more about is short for Symbiotic Community of Bacteria and Yeast. So this community is always of an unknown composition. So if you buy kombucha from the grocery store, the company that's making it probably doesn't know exactly what microorganisms are in it, but that is not a bad thing, because kombucha is a very safe food because of the acid content. And if you do production correctly, you won't have any issues. But there are specific niches that need to be filled in this community that will produce the right product. So this picture is showing kombucha in a little bit more detail. There's the layer of new SCOBY, the carbon dioxide bubbles that will escape in this open air vessel, not carbonate yet, these brown gooey bits that are all just yeast, and then the settled microorganisms that are at the bottom. So the SCOBY as a whole actually contains all of the microorganisms in the kombucha, not just in the top layer, not just in the middle layer and the strands. It's all of them, even the ones that are motile and floating around in the liquid. So the yeasts are the ones that are going to break down the sugar initially to glucose and fructose that will also feed the bacteria, and they produce the carbon dioxide. Acetobacter species are bacteria that are fed by the yeasts, and they form the acids. So big hint in a lot of scientific names, lactobacillus produces lactic acid, Acetobacter produces acids, and that acid is for flavor, and acts as a pathogen deterrent, again, much like the acid in pickles. And the pellicle is the slimy bit that forms at the top. So the pellicle is a biofilm, much like the biofilms formed by Listeria that I study. And the pellicle here is a biofilm formed of cellulose. So this functions to protect the SCOBY and the kombucha from air, because as we know, fermentation occurs in an anaerobic environment. This is at the air-water interface and will separate the oxygen environment from the oxygen deficient environment of the liquid. And kombucha, a lot of people might not like it, might not have heard of it, but definitely I recommend giving it a try. It tastes good, and maybe even if you don't like the taste, it's kind of fun to like play with the SCOBY and kind of develop a microbiology experiment in your own house. All right, now soy sauce. Soy sauce is another two-step fermentation, but with the second step of soy sauce, there is additional culture added, unlike in kombucha. So the first step is called koji. So this is a solid state fermentation. And it starts with soybeans and wheat being cooked and ground to a mash. So that cooking starts the disruption, the breakdown of molecules. It's easier for the microorganisms to get to. And it is inoculated with Aspergillus oryzae, also known as the koji mold culture. So it is not a pure culture. It is propagated from different batches of soy sauce, a very common method of bringing things through. It contains other molds as well that will produce kind of a distinct regional profile. Again, they'll kind of work together. They'll be symbiotic and break down all of the things to get ready for the second step. The enzymes specifically released by these molds are breaking down nutrients into smaller bits for the upcoming yeast and bacteria to use. So amylases will break down starch to sugar, and proteases will break down big proteins to small amino acids. So these smaller nutrients are more available. Like we talked about, food being really important for bacteria. Some of these bacteria can't break down the big starches by themselves. They can't break down the big proteins by themselves. So having the mold to create this environment where those nutrients are available really allows the bacteria and the yeast to thrive in the next step. So the next step is called moromi. And this step has water and salt added to the mash. It looks like some refried beans. And this step is where the live culture is added again, but from the second step. So this has yeasts that generate acids and other flavor compounds, and lactic acids that convert those smaller sugars into other organic acids as well. So these are just some of the many flavor compounds that all come together to make soy sauce. There are 5 different flavors that the human tongue can taste: sweet, salty, sour, bitter, and umami. Umami is kind of that meaty, mushroomy maybe taste that kind of is a hot button topic in like the foodie world. But it is actually attributable to glutamic acid, which is made by the lactic acid bacteria in soy sauce. And then to finish the soy sauce, that mash is refined by filtering. By the end of fermentation, the mash is really kind of dry looking, but that gets pressed out and then you end up with soy sauce. So bread and baking. Flour, water, salt, and sugar are things that you definitely have in your house, but something that you might not have is yeast. Yeast here is a pure culture of Saccharomyces cerevisiae, and it ferments the sugar and carbs in flour to kind of generate carbon dioxide, generate different flavor molecules. This picture here I included because you can see that the yeast is bubbling. That's how you know it's alive. If you start making bread and you let the yeast kind of bloom and it's not bubbling, you're probably not going to have very good bread. You're probably going to end up with hot Play-Doh because there will be no rise, there will be no carbon dioxide to facilitate that breadiness, right? So when you bake the bread, the proof allows the yeast to form that carbon dioxide within the dough, and then you bake that bread and the gas expands. That results in this fluffy texture that we see. There's a lot of holes. That's a place where there were yeast once. However, the yeast is killed during the baking process, and any other organisms are killed if there's anything weird in your flour or on your counter that you picked up while you were kneading. So bread is, again, a safe product, but that's because we baked it. Another thing that I wanted to note is that different yeasts will have different properties. So these packet yeast varieties are, they're basically pure culture. They're very reliable. They've been dehydrated. They'll activate quickly. But people like to use natural yeasts, like in sourdough starter. I don't know if anybody has ever worked with one, but they're kind of fun. You have to feed it perpetually and discard it. It's kind of like a pet. I'd say a little bit higher than a plant, a little bit lower than a fish in terms of effort. But then you can make a different kind of loaf with a different flavor profile. There's a different funkiness that's developed by using sourdough. And clearly, as you can see, the structure of the crumb is different here, and the crust is a little bit crustier, if you will. So the different yeasts will have a different effect on the final product, even though the yeast themselves don't end up in the final product. Now, with all of this talk about good bacteria, I thought that we should mention probiotics. Probiotics are good bacteria that are linked to improved digestion and overall health. Now, there are a lot of products out there that claim to have probiotics. There's bars and soda and peanut butter, cereal, regular butter, ice cream, and juice, and so many more. These are the ones that I thought were a little bit interesting to contain probiotics and to be labeled probiotic, but they may or may not have any actual health benefits. So I encourage your own exploration. I encourage you to read your own materials, look for reputable sources that were not funded by the kind breakfast probiotic company. You know, a lot of companies will kind of contribute to science to make the science look good for their product. But probiotics are something that you don't really need to do that with. Again, listen to your doctor if you're prescribed a probiotic, because probiotics have been linked to good improvements in health. If you do have dysbiosis, which I mentioned earlier, is the kind of microbiome being out of whack. But probiotics may not be so helpful if you are healthy. However, eating fermented foods daily will keep your gut healthy. You'll be contributing good microorganisms. You'll be adding that, letting them cycle through your body. Obviously, we've learned that microorganisms can break down things that we might not be able to. And also eat fiber, because fiber just feeds the bacteria that are already there. So you can do whatever you want, you can buy whatever you want, you can eat whatever you want. But with probiotics, I definitely say be skeptical of the products that are labeled that, and maybe just eat some pickles. So, trying this at home, I have a couple of recipes here that I personally have tried that I really enjoyed. The first one is how to make your own SCOBY. So this one's really simple. All you need is tea, sugar, and a bottle of plain kombucha from the grocery store. It gives really good instructions on what materials to use, what materials to avoid, and how the whole process should look and troubleshooting. The yogurt, again, really simple. You basically just need milk and yogurt. And bread, need flour, sugar, water, and yeast. This is a quick bread also. The recipe comes from King Arthur flour, which is a really good recipe, really good flour. And this bread itself takes only 2 or 3 hours total. So if you want to get invested in bread, but like not too invested, I definitely recommend trying this. And these are just some more references, some things that I referenced in discussion of these things. And at the bottom here, there are more home fermentation resources if you want to get into your own pickling, your own preserving. These are all from university extension resources. So they're reputable. If you want to kind of go off on your own and find more things, please feel free, but these are definitely good, reputable places to start. And any more questions, comments, or concerns about bacteria, fermentation, or anything else we talked about?
Yep, so there are 2 questions in the Q&A box. I also just wrote in the chat box that if I can have you email me those links you just referenced, I can email them out to everybody.
Yes, definitely.
So Adrian asked, what do people do with the leftover dry material after pressing the soy sauce?
That's a really good question. I didn't delve very deep into that. I'm assuming that they sell it or give it away as animal feed. A lot of these kind of spent grain products are still very nutritious and useful in agriculture in a lot of different contexts. I know that a lot of brewers and fermenters will give their spent products to hog farmers or cattle farmers. Mostly hog farmers, because pigs will eat anything.
Nice. And we have another question. I do want to remember, if I remember correctly, you said that you're going to do a Jeopardy.
Yeah,
with everyone. Okay. So everyone hang tight. We're going to do this question, and then we're going to go to Jeopardy. I thought that, so Alex is asking, I thought that one got shortbread with no yeast, not hot Play-Doh.
Okay, yes. The hot Play-Doh comment was in reference to my own experience when I tried to use dead yeast and I used a yeasted bread recipe. There are other breads that are called breads, like quick breads, very commonly banana bread, like blueberry bread, zucchini bread. Those breads use baking soda as a leavening agent or baking powder rather than the carbon dioxide produced as a byproduct of yeast fermentation, if that answers your question.
Great. And Eva had written in the chat box, I don't know if you saw it, that she has looked through a microscope at mold and taken some pictures, and she thought you described it perfectly.
Thank you.
Yeah. So before we go to the Jeopardy game, because I know some people may have to get off at, we might run over time today since we started late. I'm going to launch just my normal feedback poll just so we can get the answers before we go to Jeopardy. So if any of you need to get off hard at 2 o'clock, we understand we got started late. But if everyone could just take the poll that I just launched first, then we'll go to the Jeopardy game. And we'll give it about another 30 seconds to get more of you to chime in here. Now, there's a couple of you who haven't taken the poll yet. If you could, that would be great. Give it about another 10 seconds. All right, let's get to Jeopardy.
Awesome. All right. So do you guys drop in chat or something? Would you guys rather play for fun without teams or with teams?
Let's see.
Okay, perfect. Without teams. Excellent. All right, so I will share the Jeopardy game. I will run clicking all of the questions. If I can just get it up. Oh. All right, we'll figure it out. All right, so can you see when I go over to Jeopardy? Okay.
Oh, cool. Yeah, there you go.
Okay, cool. So this is Jeopardy. Lauren, will you be able to read out the chat answers?
Yep.
Awesome. All right. So for this, we have foods that are fermented, symbionts, places, I've got your back, the Yeasty Boys, and FATTOM. All right. So whoever wants to go first, pick a dollar amount in a category.
Yeah, whoever gets, okay, so Adrian says FATTOM for 1,000.
For 1,000, going way high. I like that.
Deep, Adrian.
All right. So in the absence of this, microorganisms will not thrive. In its presence, spores can germinate and grow.
So Alex says oxygen.
That is incorrect.
Adrian says pH.
Incorrect.
KL says moisture.
That is correct.
All right, KL.
All right, let's get back here. All right. Whoever wants next.
All right, someone give a category and a dollar amount. Okay, KL says FATTOM for 800.
All right. This needs to be kept in delicate balance for the human body to survive, but fermentation can occur at a greater range.
KL says pH.
That is correct.
So does Adrian. You guys almost got in there at the same time.
Excellent.
Okay, someone else pick a category and an amount. Okay, we have FATTOM for 600.
All right, we're just going down. A scale of 1 to 14. The closer you get to either end, the more challenging it is for microbes to thrive.
We got a lot of pHs coming in.
Again, correct. Oh, wait, okay. Hang on. Okay, we got it.
Who's gonna pick a category, a dollar amount? Anybody who hasn't chimed in yet? We have Eva says foods that are fermented for 1,000.
All right. Produced like a pickle, this condiment will add an acidic but spicy kick.
Adrian says soy sauce. Alexis says hot sauce.
Hot sauce is correct.
Sauerkraut.
Ooh, sauerkraut. I wouldn't call sauerkraut spicy, but you know, you could add whatever you want to it.
But you were going for hot sauce?
I was going for hot sauce. Yep.
All right, next category, dollar amount. Who wants to weigh in? Places, 4,000.
Places. All right. So organisms turn sweet tea into carbonated and acidic kombucha while living in this acronymized collective. So I'm looking for the place the organisms are.
Levi says SCOBY.
Yes. Excellent.
Nice. All right. Next category, dollar amount. Levi says places for 800.
Places for 800. Bacteria cannot undergo fermentation in this environment, but yeasts can.
Alex says oxygen.
Yes. That is an aerobic environment which contains oxygen.
All right, someone pick a category. Category and dollar amount. Anyone? Anyone? Foods for 400.
Foods for 400. Stereotypically enjoyed by mice, this food can develop into its final form with help from bacteria, yeast, or mold.
Oh, we got lots of cheese coming in.
Very good.
I can't even keep up. All right, how about Molly? You pick the next category and amount. Food for 200, she says.
Food for 200. This food is a preserved form of fresh veggies which have been lacto-fermented.
Pickles.
Pickles,
Sauerkraut, pickles, 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 pickles. Guys, you're making me hungry for pickles.
Yeah, I know. That was a
Alexis, why don't you pick the next category and topic? We'll let Alexis choose. And I do have a jar of pickles in my fridge that I would eat, but I cannot get it open. It's so tight. So, so Alexis says bacteria, what is it? I got your back for 400.
I got your back for 400. Cheese and yogurt would both be impossible without the addition of these bacteria.
Got probiotics, lactose, cultures.
Cultures is what I was looking for. Starter culture makes cheese and yogurt possible.
All right, KL and Ruby both got that. Ruby, why don't you pick the next category and amount? Symbionts for 600.
Symbionts for 600. In kombucha production, this is what is formed by the symbiotic bacteria that manufacture cellulose.
You may have stumped.
All right,
for this one, I do the Jeopardy music?
We can't.
Oh, we have SCOBY.
SCOBY's good.
Two people, both Adrian and Levi, have said SCOBY. Is that correct?
What I was looking for was pellicle. I would have accepted biofilm. SCOBY, as I mentioned, is all of the bacteria, not just the bit that's formed on the top. Fun fact.
Okay, Levi, why don't you pick the next topic and amount? Let's see what comes up. Oh, thank you. I wanted someone to pick the Yeasty Boys. So Yeasty Boys for 600.
All right. In the SCOBY, this term is used to describe the air-water interface of the pellicle. Hint, it's also what I study.
Biofilms?
Yes, absolutely.
All right. Eva, good job. So you pick the next one. And I think we'll do like 3 more because I'm looking at the time. We're going to have to wrap up. So we'll do this one and then 2 more questions. So Eva, you get the topic and amount this time. So she's going for the Yeasty Boys for 1,000.
Yeasty Boys for 1,000. Yeast will still ferment when this gas is present.
Oxygen.
Correct.
Both Alex and KL went in there. So Alex, you got in there first. Why don't you pick the next topic and amount? Places for 400.
Places for 400. This type of oxygen-lacking environment is where fermentation typically occurs.
Ooh, anaerobic.
Yes.
Both Alex and KL. Yeah, that was fast. You guys type fast. All right, so this is our last question. KL, you pick the topic and amount. This is gonna be our last one. Make it a good one.
Oh, I hope it's a good one.
FATTOM for 200.
FATTOM for 200. Excellent choice. Nutrients that are available to microorganisms are known as this.
KL says food.
Yes, food, absolutely.
Nice. You guys rock this. What a great little fun game. Emily, thank you so much. Before we go, are there any final questions for Emily today? Well, that was such a good idea. Ruby, are you talking about the Jeopardy game? Because I agree with that. I thought this was such a great way to engage you guys.
Thank you.
Yeah.
Oh yeah, you can see my shirt now.
Oh, there you go. I love pickles. Well, that's great. Well, Emily, thank you so much for sharing your work with us today. Thank you all for joining us today. Hopefully I'll see you next week at the Natural Resource Management Academy or the following week when we bring our veterinarian in. But again, thank you. And again, I'll have Emily share all those resource links and I'll send them out to everybody once I get them, okay? So you all have a great rest of the day and we'll hopefully see you all soon. Bye, everyone.